Alrighty, today we are going to be talking about the chemoreception of smell and taste. We are going to be talking about hearing, and we are also going to be talking about vision and how the nerves and action potentials in the eye work together so that you can tell the difference between light and dark and communicate that to your brain such that you can see images. So, the first thing we're going to be talking about is taste. Like I said earlier, my Apple Pencil is not working very well, so you're gonna have to bear with me with these drawings. So, in a nutshell, if you were to zoom in on the surface of your tongue, you're gonna see these little raised edges, and these are your taste buds. And beneath these taste buds are these long receptors, and they're almost kind of shaped like something like this. And beneath all of these taste buds are these little tiny nerve receptors that all have different mechanisms by which they receive input. And so these um, each of these receptors underneath the taste buds all respond to different chemicals to tell your brain that you are tasting something different. I'm going to zoom in over here. And first, what we're going to talk about, oh, that's a little large, is salty. And so the way that your brain tastes salt is in response to two different ions, namely sodium and potassium. And so what we're going to see here is as salt enters in through these little receptors, sodium is going to induce an action potential. And that's going to increase the overall positivity on the inside of this specific receptor cell. When this receptor cell opens, it's going to activate the calcium on the inside in the smooth endoplasmic reticulum of this particular cell. And what it's going to do is it's going to increase um, the overall charge on the inside of the cell. And when that happens, it's going to cause the potassium leak channel to open, and that's going to cause potassium to rush out of the cell. And during this change of voltage, it's also going to cause this voltage gated calcium channel to open. And as you can see here, it has its two little gates. And so when it's active, what it's going to do is this action potential will increase the positive charges, which is going to cause these vesicles to release all of their little um, all of their little chemicals, all their little neurotransmitters on the inside. Now um, we're not exactly sure what neurotransmitter is released through these taste receptors, but what we do know is that when it it activates a response through a neuron. And so that neurotransmitter is going to be released. And my Apple Pencil's not doing well. I'm gonna need to buy a new one. But anyway, so through this process, this is how your brain tastes salt. And so it's actually through the induction of sodium itself. The sodium channels are actually the one that depolarize these specific salt receptors and induce an action potential. Now, when we go over here, we have something called the ENaC channel. Okay, the ENaC channel. It's very important. It is a specific type of voltage-gated sodium channel, and these ENaC channels are also sensitive to hydrogen. Okay, and so when you taste something sour, you're actually tasting the presence of protons on the surface of your tongue. And so this presence of protons on the surface of your tongue is going to increase the overall positive charge, similar to how the taste of salt worked. It's going to increase the overall positive charges on the inside of this cell. And what's going to happen is it's going to close this potassium leak channel. And that's not going to allow any more potassium out. And so you're going to get this really positive cell. And what that's going to do is increase the concentration of hydrogen on the inside of this cell. And when you have this increased concentration of hydrogen on the inside of the cell, what you're going to get is also, again, a response that's going to go down and tell the brain, hey, I am tasting the presence of protons. Now, sweet, bitter, and umami all are different. So, as I wrote here, erase this. Each of these different flavor profiles have different. They all have different G proteins that respond to the presence of these different flavors. So, for example, if you're tasting something like glucose with sweet, bitter, and umami, um, yeah, sweet is carbohydrates, bitter is calcium, and a molecule called quinone, and umami is in response to glutamate. Because each of these are all different molecules, each G protein here is going to look different between sweet, bitter, and umami. So each of these different G protein coupled receptors, this is what this stands for. G protein coupled receptors all respond in a different way to the presence of one of these three molecules. Now, for example, let's say this little guy represents a G protein here. Okay, we have our alpha, our beta, and our gamma subunit. What's going to happen is, let's say we have glucose. So we have glucose, which is H6C3O6. Okay, so glucose is going to bind to this G protein coupled receptor, and the gamma subunit of this G protein coupled receptor is going to bind to something called phospholipase C. So phospholipase C is a enzyme that is responsible for the what's it called? The cleaving of a molecule called IP3. So phospholipase C is going to cleave that molecule and release IP3 into the interior of the cell, and IP3 is going to go along and bind to the sarcoplasm, or excuse me, not the sarcoplasmic reticulum, the smooth endoplasmic reticulum. And when it goes to the smooth endoplasmic reticulum, the ryanodine receptors are going to release calcium from the inside, as we know, because the um, endoplasmic reticulum is obviously a calcium storage site. It's going to open up those receptors and release calcium. And when calcium is released onto the inside of the cell, that's going to change the overall charge of the cell. So we're going to have a hyperpolarized environment on the inside of the cell. And that is going to increase the driving force of potassium outside of the cell, and in hopes to reestablish some normalcy in the charges from the interior to the exterior of the cell. Um, okay. Then what's going to happen is when the calcium on the inside of the cell increases in concentration, it's going to cause these little vesicles full of neurotransmitter to be released. And so what we're going to have is, in this case, glucose is going to cause an increased release of this neurotransmitter, which is going to tell the brain, hey, I'm tasting something sweet. And same goes for the two molecules for bitter and sweet, namely calcium, quinone, and glutamate. So now we have those guys all out of the way. Now, fats, they follow a similar um, path. We're not too sure actually how it works yet, but it is responsible in lipids. We imagine it's something similar to a G protein coupled receptor response. Okay, so that is taste. So now we're going to focus on smell. So here I've drawn a little guy. You can see his nose. In this little highlighted region up at the top of your sinus cavity, you have something called your olfactory bulb. And in this region of the olfactory bulb, you have a bunch of cells called olfactory epithelium. And these olfactory epithelium are connected to olfactory nerves, which go through something called the cribriform plate. Um, Now, the cribriform plate is essentially a massive bone that these nerves go through. And unfortunately, if you shear or break the cribriform plate, you'll shear the nerve endings that go through it, and you won't be able to smell anymore. However, on the area below the cribriform plate, these olfactory neurons can actually be regenerated over time. And so they're recycled similar to other cells or nerves in the body over time. And that's how you are able to continue to smell. However, if you are ever in like a traumatic accident or something, that cribriform plate is sheared, it is very, very likely that those olfactory neurons will be severed and you will no longer be able to have your sense of smell. So, as these neurons ascend below, or excuse me, descend below the cribriform plate into the olfactory epithelium, there is a thin layer of mucus that actually surrounds. That's why it's extremely important that your nose is wet. That's why if you see dogs lick their nose, it's to help enhance this process. Because in order for you to smell something, the molecules that attach to this olfactory bulb have to be dissolved in solution. Now, the dissolution of these small molecules happens very quickly. Um, but that's how you smell. So you can't smell, quote, a like dry substance. So the molecules themselves actually have to be dissolved in this mucus layer in order for these olfactory neurons to pick up on the signal and be able to relay that to the brain. So similar to as we talked about with the your sense of taste, there is a process um, by which these molecules that you're smelling actually trigger a reaction cascade in the olfactory bulb. And so right here, you can see this little oddly shaped kind of barbell looking symbol. This is going to be, for example, a molecule that your brain is going to smell. So on the surface of these olfactory bulbs, you have a molecule called adenylyl cyclase. And adenylyl cyclase is responsible for a conformational change that, when activated, turns AMP into cyclic AMP. So you'll see that transition from AMP to the well-known CAMP, as we see through many different physiological processes in the body. And so when AMP is changed to cyclic AMP, it is going to cause the opening of a symporter, as we talked about at the beginning of the year, a sodium and calcium symporter. And when this symporter is activated, we're going to see a rush of calcium and sodium into the olfactory bulb. When this increase in concentration of sodium and calcium on the inside of the cell results, it is going to push chloride against its concentration gradient out of the cell. And that is essentially how the brain is going to, this change from negative to positive is what is going to transition, kind of like what we talked about with length constant in other neurons. Um, it is going to trigger a action potential into the olfactory sector of the brain, telling your brain what you are smelling. Okay, now we are going to talk about hearing. So the ear is very unique. It operates due to the external process of the ear and a small channel that guides you down to your tympanic membrane, which is also known as the eardrum. Now, behind this tympanic membrane, which I'll highlight here, there are three bones. The malleus, the stapes, and one other one that I'm totally forgetting off the top of my head. It's not important for this at the moment. However, when the tympanic membrane moves against these three bones, they relay the frequency and the speed of the vibrations. And so, because as we know, sound waves is just compressed air. And so this compressed air will travel down the ear canal, creating vibrations, which is transduced through these bones, which I'm gonna highlight the main three right here. So we got this guy, and this guy. And so when that air is compressed and creates those little vibrations, it is going to travel through the tympanic membrane, move against those bones, and that vibration against the cochlea is going to cause a vibration in the fluid that fills this inner ear section. And these are called the ciliae vestibulae. They're like little hair-like structures that will vibrate in the presence of this compressed air. And so, what we're really going to focus on for the moment is something called the basilar membrane. And this is this little channel that you can see that sits below. Get a different color to highlight it. It sits below the cochlea right here, in between. And if you were to basically roll out that basilar membrane, it's going to look something similar to this diagram I have pre-drawn over here on the right. And what you're going to notice is that as you extend down or to the peak of what would be that little cochlear shaped um, portion of your ear, is you'll see the width actually increases. And the reason that's important is because the fibers that what would be the right word for this, that cross from one end to the other are shorter on one side and longer on the other. And the frequency at which those vibrate changes because of that length. And so you'll see on the shorter side, you're able to detect sounds of about 20,000 hertz. And so that's where you're going to get your really high pitched noise. And so the reason that's important is because in this section, the reason for example, like let's say your grandparents, the reason your grandparents struggle so much in hearing you is, especially like young children, is they have higher voices. So oftentimes the problem is that they really struggle to hear high frequency noises. And that's because these these fibers, these that descend across this basilar membrane, all sound has to travel past those to get to the areas of lower frequency. So if that doesn't make too much sense, let me explain it in a different way. Basically, this is like the beginning of the trail that sound has to travel. So even if it's a low frequency noise, the the sound has to travel past these or these high frequency regions first in order to resonate in the right region of the cochlea. And so over time, those high frequencies will just get worn out. They won't be as responsive to those high frequencies. And so when you see somebody with hearing loss, especially normal hearing loss that just happens over old age, it's just because those fibers have been slowly worn out over time and they're not as responsive to those high frequencies. Okay. Um, let's see what else. Okay. So now what we're going to do is we're going to zoom in here even further on the basilar membrane. And we're going to have this little diagram drawn that shows a few different areas of the inner ear that is actually how the action potentials are transduced to the brain for hearing. Okay. Am I forgetting what this stands for? Okay, so here we have something called the tectorial membrane. I'm gonna color it in here so it's easier to see. Okay, and as you can see, these little ciliary hairs um, actually descend through the endolymph. They're connected in this other region called the perilymph over here. Um, and they descend through that region and they poke through with these little hair-like finger projections and they just barely slightly touch the tectorial membrane. And as the tectorial membrane vibrates, it's actually going to push on those hair endings, and that mechanical action of the movement of the hair is going to cause an action potential. We'll talk about it a little bit more in detail. As you can see, there's a couple other things I want to point out. Number one is that the perilymph is very low in potassium, and the endolymph is very high in potassium concentration. Now, the perilymph being low in potassium concentration means that it is also at the same time higher in sodium concentration. And that's going to be important as I explain how the different mechanical channels open, um, causing that change in a, gradient potential, which is going to be how you induce hearing. Okay, so actually, how should I explain this? Let's do it like this. So if we were to zoom in here on the endolymph, what we're going to see is that there is a very high potassium concentration on the outside. And we're going to have a bunch of these potassium and sodium symporters or ATPase transporters that are going to move potassium either with or against its concentration gradient, depending on the need. They have a special name in the ear, and they're called NK2C pumps or NKCC pumps that are going to be responsible for this movement. Now, let's zoom in on the cilia over here. So this is essentially what the cilia looks like under, if you were able to magnify it. What you're going to see is at the top of these little finger-like projections, there are some that are taller and some that are shorter than the others. And in between each of these fingers is collagen fiber. And these collagen fiber, they're actually connected to these mechanically gated um, potassium channels. What you're going to see is as these hairs bend, let's say it bends this way, it is actually going to pull on these collagen fibers that is going to change physically and mechanically alter the conformation of these potassium channels, allowing for an influx. As you can see here, the endolymph is very high in potassium. It is going to cause these these channels to be opened, and the potassium is going to rush into the interior of the cell, completely altering the all of these negatives over here. Because as we know, the concentration on the inside is slightly negative compared to the as far as the perilymph to endolymph goes. And so the mechanical triggering of these mechanically gated potassium channels opening is going to cause potassium to rush into the cell, changing the resting membrane potential of these cells, which is going to activate the release of these voltage gated calcium channels and voltage gated sodium channels. And so this change from the endolymph to the perilymph, oh, that was the wrong tool. From the endolymph here to the perilymph here, that changing of charge is what's going to cause the um, brain to understand that sound is present and will be able to interpret it. Okay, now we're going to talk about the eye. So here in this dark spot right here, we have pre-drawn out. This dark area is the macula densa. So when you hear the eye doctor maybe talk about macular degeneration, it's just this the reason of very dense cells where the lens of the eye focuses on to transmit um, light into action potential so that your brain can, tell light from dark, um, colors, edges, things like that. Some macula densa. So here in our lens, as you can see, connected to the lens is something we call suspensory ligaments. And the suspensory ligaments are what are going to either contract or relax to change the shape or the convexity of the lens to direct light in a certain manner in which allows you to either see light close or too far. So basically just changes the angle at which that light is reflected, which you can see at different points. Um, this area is obviously all full of vitreous humor. We're not going to talk about that too much. But what we're going to see is as these ciliary muscles, so I can get my pen to work, as they relax, they're going to straighten out, which is going to cause the lens to actually become more circular. Now, if they tighten, so let's say these muscles contract, it's going to cause the lens to look more and more oval, like that. And so essentially based on the contraction or the relaxation of these ciliary muscles, you are going to be able to change the convexity of the eye lens, which will change where light is directed onto the macula densa. Now, at the back of the retina, we have a bunch of cells. Now, these cells are well known. You're usually taught about it in middle school. You have your rods and your cones, but something we're going to talk about that's important is goes beyond the rods and the cones. So the rods and the cones are the bottom cell to basically a column of cells. So above the rods or cones, connected directly above them, you have something called a bipolar cell. And the bipolar cell has a unique effect on communicating different neurotransmitters to the brain about telling it whether or not it's seeing light or dark. And then connected to the bipolar cell is something called the ganglia cell. And the ganglia cell also has a role in changing neurotransmitter release to tell the brain whether or not it's seeing light from dark. So here I have drawn up a rod and a cone connected to a bipolar cell, which is then connected to a ganglia. And I'll talk about how the eye communicates light to the brain. So here, as you can see zoomed in at the bottom of oh, let's see, at the bottom of the cone cell, we have another G protein coupled receptor. Now, the unique thing about this G protein coupled receptor is inside of the receptor, there is a molecule called 11 cis retinol. And 11 cis retinol is unique because it has as is mentioned in organic chemistry, it has a cis tail on it, meaning that it has almost like a curve to it. And this 11 cis retinol, when hit with different nanometers of light wavelengths, the light actually has the ability to sever this double bond here. And when this double bond is severed, it looks like this. And it actually changes it from 11 cis retinol to 11 trans retinol, meaning that that tail end of the molecule is actually straight. And when that molecule is hit with light and it breaks, it causes a enzyme called phosphodiesterase, or excuse me, when that 11 cis retinol becomes 11 trans retinol, it releases the alpha subunit of the GPCR, this G protein coupled receptor, and it connects to this phosphodiesterase enzyme. And when this phosphodiesterase enzyme is activated, it comes over here to this this would be a ligand gated sodium channel. This ligand gated sodium channel is activated by cyclic GMP. So normally this cyclic GMP molecule right here causes this sodium channel to remain open. There's a little gate, essentially. You could call it a little gate that remains open, allowing sodium influx into this cell. And when the phosphodiesterase is activated, it actually turns the cyclic GMP into, oops, the wrong tool. Turns it into GMP. So essentially, it just removes the cyclic portion of that molecule. Um, and when that happens, this arm here is going to close. It's going to look like this. And when that's closed, there's going to be no longer a release of sodium into this cell, which is going to inhibit the release of glutamate. And so in the brain, the primary neurotransmitter is called glutamate. And glutamate is going to be important here even more in a minute. Glutamate, all the green, even though I wrote it in red, but you get the point. So glutamate is released less, and what happens when it's released less is it causes these voltage gated sodium channels, or excuse me, voltage gated calcium channels to close. And when the voltage gated calcium channels close, they're not able to bind to these vesicles, releasing more glutamate. And so what you're actually going to see is in the case of light, even though this seems a little like a little backwards, what you're actually going to see is in the case of light, there's actually less action potentials being released from the from this bipolar cell. And so at first you might think, oh, well, less action potentials means the brain will see dark, but that is actually not the case. Less action potentials in this case is going to cause the AMPA channel on the ganglia to tell the brain that it is seeing light. So it's a little interesting situation going on here. So let's say the reverse happens. Let's say we have no light that hits this G protein coupled receptor. Well, if we have no light that hits this G protein coupled receptor, I'll use my little pointer. If we have no light that hits the G protein coupled receptor, that means this alpha subunit right here, boop, he's not going to get turned on, which means he's not going to activate the phosphodiesterase, which means he's not going to change cyclic GMP into GMP, which means this will work, that this is going to stay open, which is going to allow sodium to rush into the cell. When sodium rushes into the cell, it's going to cause these vesicles, or it's going to cause calcium to rush into the cell, which is going to bind to these vesicles, which is going to release more glutamate, which is going to bind to the mGluR6 G protein over here. And that is going to cause this calcium channel to stay open, which is going to cause more calcium to rush into the cell, which is going to connect to these vesicles, which is going to result in increased glutamate in this synaptic cleft here. And in this synaptic cleft, the brain will now tell, or the ganglia will now tell the brain in the presence of increased glutamate concentration that now the brain is seeing dark. So that's a rundown of how the brain can see light from dark. Okay, so now we're going to talk about something that is extra fun. So in each photoreceptor, each photoreceptor is part of something called a photoreceptor field. So each rod and cone cell that is connected to a bipolar cell, that is connected to a ganglia, is all part of basically like a little group of cells that all work together to communicate levels of light or dark that the brain is seeing. And so in each photoreceptor, you have a group of cells that are going to be something we called on center, and then a group of cells surrounding them called off surround. And I'll explain a little bit more what that means in a minute here. Then beneath each of those on and off center, you have something um, called horizontal cells. And these horizontal cells run next to the rods and the cones, obviously running horizontal to them. And they release something called GABA. And GABA is a inhibitory molecule that basically causes a string of IPSPs or inhibitory postsynaptic responses to change the rate at which these the neurotransmitters are released to deliver varying signals to the brain, which helps you see varying intensities of light, helps you identify shapes and edges, um, all in the hopes to help your brain understand the world around it. So, below these horizontal cells, or excuse me, I guess above, closer to the lens, you have bipolar cells. And depending on the cell, they will either have the mGluR6 receptors on them or AMPA receptors. And then down here's the ganglia. Okay, so let's get a scenario going on here. So let's say we've got our off surround. We'll use purple. So we'll use our off surround over here, and it's releasing glutamate. Man, this thing is terrible. Okay, all this purple is glutamate. We've got our on surround releasing glutamate. We have another off surround, or excuse me, on center. And we have our off surround releasing glutamate. And so the important thing to remember is that all photoreceptors will always release glutamate. Now, the rate at which they release glutamate obviously will depend on if they're on or off surround and will depend on the horizontal cells GABA or um, their quantity of release of GABA. So let's say we have light. And this particular little stream of light, you could say, is going to hit all three of these regions of the on and off surround. All right, light up here. Oh my gosh. This is bad. This pencil is not working. Okay, whatever. That says light. So light is going to hit each of these off, surround, and on center photoreceptors. Now, the reason they're called off surround or on center is because of what they do to the bipolar cell following it. So, in the case of off surround, let's look at that first. In the case of off surround, we have a off surround photoreceptor, and light is going to hit the off surround photoreceptor, which means this is going to cause a decreased release of glutamate, right? Because it's off surround and it's being hit by light, so it's going to release less glutamate, and that they're going to the photoreceptor will then communicate that to the bipolar cell. And the other thing, so one other thing I forgot to mention, unfortunately, before, which would make this make even more sense, is the center photoreceptors, whether they be on or off center, are going to contribute approximately. The easiest way to explain it is to give it a value out of a percentage. Now, it doesn't actually work this way, but it's the easiest way to conceptualize it. So the on center is the on or off center will contribute about 80% of the overall action potential response of a photoreceptor field. And the off center will each contribute 20%. So 80 and 40, 100. Wait. Excuse me. Yeah, I totally said that wrong. The on center is 60% and the centers are 60% and the surrounds are 20% each, equaling 40, equaling 100. Sorry about that. Okay, so where was I? So for example, if light hits this on center, so this is on. So we're gonna have light hit here. We'll put a value of 6 to 2, 60%, 20%, 20%. Now, the on center in the response to light is going to contribute 6 action potentials to the overall to the bipolar cell. So this, because it's on center and it is being hit by light, is going to tell the bipolar cell, hey, I'm being hit by light. So it is going to increase its glutamate response. And the bipolar cell will be able to pick up on that, say, hey, we have an increase in glutamate being released from our on center photofield, and it will compute and it will communicate to that to the brain. And the same as I explained earlier will happen with this other off surround photoreceptor. Okay, now in response to glutamate, these are all going to I'm going to erase this. These are all going to bind to the mGluR6 pathway. Um, Let me actually look real quick because I'm forgetting something. I know. Oh yeah. Uh, off. So AMPA. Yeah, so it would be mGluR6. Okay, so in this case where we have on center and off surround, that is when these are going to bind to the mGluR6 pathways. Now, in the circumstance of having off center but on surround, that's going to totally change this, right? And so in the case of off center, the actual bipolar pathway is actually going to change to an AMPA pathway instead of an mGluR6 pathway. Um, okay, one second here. Bipolar AMPA, GABA's activated, GABA's inhibitory. Hmm, what is GABA doing in here? On center, off surround, GABA. All right, Aaron. Well, if you end up actually listening to this one part, I'm sorry, but I cannot remember what GABA does or exactly how it interacts. But I do know that GABAtory releases IPSPs, which is going to decrease the rate at which glutamate stimulates the bipolar cell. But I can't remember much past that. Anyway, so that is how the brain sends those signals in a nutshell. So I'm going to show you another way to explain it using this lovely diagram. I'm going to shrink it actually so that it's easier to see. Okay, so these two circles are going to represent two different photoreceptor fields. Okay, so let's say for the time being, we have an on center, off surround photoreceptor field. And let's say that we have light that comes in and it hits the entirety of the photoreceptor field like so. What this is going to result in is, as I explained earlier, we're going to have 60% of our action potentials come from the on center and only 20% on either side come from our off center. We're going to only see a result in now, right? 6 action potentials. So we'll draw a little diagram. Looks like this: 1, 2, 3, 4, 5, 6. Okay, Okay, 6 action potentials. That's because this is an on-center photoreceptor, meaning that when light hits it, it is going to tell the brain, "Hey, I'm seeing light." But the off-center are being stimulated by light, but they're telling the brain, "Hey, I am not I am not doing my job, basically. I'm doing the opposite of what I should be doing." So it is not going to release action potentials. It's going to decrease the rate at which glutamate is released to tell the brain that it is not seeing light. So that's why you're only going to see 6 action potentials from this. Well, now let's say we have light that only hits, let's say, this section. So now we only have light that hits half of the photoreceptor field. Well, now this, right, this is an off center and it is receiving light, so we're not going to receive action potentials here. Okay. And we are on center, so we are going to receive action potentials here. And we're off center here and we're not receiving light, so we actually are going to receive action potentials here even. So now this action potential wave is actually going to have 2 more action potentials in it. It's going to receive a total of 8 action potentials. Okay. We'll give you an let's do another example. Let's do let's say we have off center, off surround, and we do receive light. Well, now what's gonna happen? Well, as you guessed it, 10 action potentials. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And that's because this specific photoreceptor field is primarily designed for telling the brain that, hey, we are not receiving light. So if we are receiving light, that is actually going to cause um oh wait, off, off, off. Oh, nope, I did that wrong. This is going to be, it would look more like this. There we go. Okay, got it aligned here. We're actually gonna have no action potentials, right? Because this part is telling the brain, hey, we're not receiving light, and we are receiving light. So there's gonna be a release of 0 action potentials from the brain to help communicate that idea. So there you go. There's photoreceptor fields, vision, bipolar, cone cells, horizontal cells, rod cells, hearing, vision, and taste and sense of smell.